Thank you. Hi, everyone. I am Reetika Ghag, and today I'll be presenting my work in Dr. Molla's lab titled scHOCMO, a higher order correlation model for single cell multi-omics. Oh, yeah. Can you hear me better? Yeah. So I'll be presenting my work, scHOCMO, a higher order correlation model for single cell multi-omics. Here are my disclosures. As a part of my talk today, I'll be covering some single cell basics. I'm pretty sure everyone is familiar. The motivation for developing the method, the meat of the method, the data set we use to illustrate scHOCMO, and finally some results and a concluding summary. As we saw in Dr. Gianma's presentation yesterday, single cell is associated with this fruit bowl, which allows us to have readouts over individual cells, whereas bulk can be compared to this fruit jam. Sorry, my pointer isn't working, which averages the readouts among all different cells. And that offers several advantages, such as enabling to study the heterogeneity, to identify cell types driving a specific phenomenon. Lineage tracing studies help us to understand how these evolve over time and understand disease progression. And finally, the stochastic gene expression studies help us to understand how genes are regulated as a state of the specific cell types. So, leveraging these different advantages and having single cell readouts through to across all these molecular layers, that enables a systems level interrogation to understand the regulatory dynamics across various molecular layers governing disease progression, and also paves the way for multi-omics data integration. But multi-omics single cell data integration is posed with several challenges, such as the mosaic data integration, wherein your data is a complete disjoint set of cells and features. As you can see, the blue box, which is a set of genes across a set of cells, and the SNA tag matching. Oh, sorry. Oh,
You can say interruption, laser, so
oh,
you
I.
can you can
Okay.
be looking here,
Okay.
so you can speak to the mic.
Sure. Thank you. And then the distinct features. So we have the gene expression, which is this blue box, and then a single cell ATAC expression, which is the orange box. And as you can see, those are computed across distinct feature spaces and distinct cells. So that does not allow us to have a common axis or anchor to create a unified integrated representation. The sheer amount of data that single cell technologies generate pose a scalability challenge. And also, existing methods for single cell multi-omics data integration, such as are mostly based on deep learning frameworks, which are inherently prone to black box problems, or unsupervised methods such as NMF or PCA, which lack the unique solution. So, in order to address these challenges, we are extending a previously developed HOCMO method by our lab to now capture the single cell dynamics. And with scHOCMO, we are trying to develop a data driven approach to inform these cell identities, independent of known gene markers, enabling the discovery of novel biomarkers, and also capture the complex regulatory relationships among multiple single cell modalities. So, just a brief background of the HOCMO model. The higher order correlation model is a non-negative tensor factorization based model for bulk data. And here we are looking at two data sets: a reverse antibody based reverse phase protein data set represented by a tensor which measures proteins treated with 6 ligands across 4 different time points, and mass spec based global chromatin profile tensor measuring histone marks treated with 6 ligands against 4 time points. And with this study, we were trying to understand the tumor microenvironment as a function of the intra and intercomponent signaling pathways, trying to find the histone marks governing breast cancer. So these tensor data sets are representing 4D data, like proteins, ligands, histones, and time points. And we conducted a differential gene expression analysis to get the log-fold change, followed by representing this 4D data as two separate tensors. Each of these tensors was then decomposed using the non-negative Candicom parafact method, which we'll view in a minute, and it results into latent factors, and these latent factors capture the hidden structures, in this case the underlying molecular signatures, like the protein signature and histone signature, which were further analyzed to understand the higher order correlations looking at the auxiliary matrices. But what is a tensor? So tensors are mathematical constructs and multidimensional arrays that scale from a scalar to vector and generalize over higher dimensions. And tensors have a property, and the order here is basically representing a dimension or a rank. And the tensors have this property of being decomposed into these latent factors, which are a lower dimensional representation capturing the hidden structures, which can be summed back to reconstruct the original tensor. So that provides a solution to to enhance the interpretability and also guarantees a unique solution. So here are some results of the SCHOCMO method, where the latent factors identified two distinct signatures and show the cluster memberships of phosphoproteins. We have ligands and time, and similarly for the histones over treated with 6 ligands over different time points. We further captured the pairwise correlations and observed an enrichment over the 24th hour, as you can see here, for both the GCP and RPPA datasets, suggesting a possible onset of breast cancer initiation for all growth factors. The higher order correlation scores now are used to understand these complex dynamics and and give the correlations among the enriched ligand, phosphoprotein, histone, histone marks, and that drives the hypothesis generation of how different proteins can be treated with ligands and affecting histone marks, resulting in open or closed chromatin. And we also used these enriched phosphoproteins to identify cell signaling pathways driving breast cancer. The promising results of SCHOCMO motivated us to extend it to capture the single cell dynamics through our method scHOCMO, which is basically broken into three parts: a tensor representation, trillion tensor pipeline, and SCHOCMO model. And here, for our use case, we'll be focusing on single nucleus RNA-seq and ATAC-seq data, which are computed across completely disjoint set of axes, often termed as unpaired. And if it is a joint assay, it's a paired assay. So the first step is the tensor creation, tensor representation step, which tries to create a unified representation of all the different modalities. And in this case, we aim to create a tensor or a unified representation of cells, genes, and peaks. And that can be achieved using three strategies. So we developed a linear regression-based stacking method, which models gene expression, Y, as a function of the peak intensities across all ATAC-seq datasets represented by X. And the coefficients capture these missing relationships that could be helpful in creating the tensor. The second is a Khatri-Rao product, which is a mathematical concept, and for a given common axis, in this case, it's the gene, it computes the pairwise correlations among gene expression and peak intensity. Finally, we developed a novel normalized append-based stacking method, which is an adaptation of the Khatri-Rao product and normalizes the pairwise correlations. Here, for a given gene or a common axis, in this case it's a gene, A represents the gene expression from, let's say, cell C1, and B is the peak intensity for a given cell C2. And we wanted to capture the effective contributions for gene expression and peak intensities in a given pair of cells. So this was the rationale driving the method. And we introduced a map-reduced based implementation since we have a column independence that helps us to chunk the input matrices into smaller blocks, and then finally create a reduced representation, and finally, sorry, reduce it to create the final tensor of genes, peaks, and cells. This tensor is further decomposed using the trillion tensor pipeline. Um, the trillion tensor pipeline chunks this larger tensor into smaller blocks, and each of these blocks is further compressed to obtain lower dimensional replicas. So the Y are the replicas, and each of these replicas is then factorized using non-negative Candy-Compare-Fac decomposition to yield latent factors, joined through a non-negatively square equation. And finally, the latent factors, the intermediary latent factors, are rotated and permutated to obtain um, latent factors A, B, and C, representing the underlying signatures of cells in A, peaks in B, and the genes in latent factor C. Finally, these latent factors are analyzed through the HOCMO pipeline to obtain the higher order correlation scores. So, latent factors ABC were representing cluster memberships of cells, peaks, and genes, which are now used to generate a pairwise correlation matrices denoted by M1, M2, and M3, which could be further used to calculate and model um, the higher order correlation scores given here. To demonstrate our method, we used a diabetic kidney disease dataset, which contains of an unpaired single nucleus RNA sequencing dataset having 6 control and 5 DKD samples, and an SN-ATAC dataset with 6 control and 7 DKD samples. The dataset identifies 12 distinct cell types, and if you look at the data scale, we are looking at more than 1 trillion data points. As a part of the pre-processing steps, we filtered for low-quality cells, removing doublets and empty cells, performed gene-level filtering, followed by log normalization and scaling. In order to create a unified axis, since we started with a complete disjoint set, we annotated the peaks for single nucleus ATAC-seq with its corresponding genes. And we also conducted a differential gene expression analysis, resulting in 608 of commonly identified DEGs that we use for our final data set. In order to understand if lower dimensional data sets are efficient in capturing the underlying biology, we ran simulations and we created simulated data sets using 2 strategies. One is a random subsampling strategy, and the second is a non-negative matrix-based subsampling. So the goal here is to downsample our data sets, and the NMF-based subsampling aims to preserve the underlying biology. So as you can see here, the simulated data sets are for snRNA as well as SN-ATAC data sets are very representative of the original study and map to all the major cell types. Here are some results. From our benchmarking analysis, we compared the tensor creation strategies for a million scale tensor. And as you can see, the Khatri-Rao product appears to be the fastest and the most space efficient. However, it is prone to a known explosion problem, which limits its use for very high dimensional data. So overall, the normalized append-based stacking method emerges to be the most robust and efficient. Secondly, we conducted a benchmarking analysis to understand system-level differences on a billion-scale dataset, and it introduces a 1.5 time delay, just attributing to the hardware differences with a mean squared error shown here. Lastly, to understand how different hardware architectures influence the performance, we conducted another benchmarking study on a million scale dataset comparing performances on GPUs, CPUs, and TPUs. As you can see, the GPU outperforms the performance compared to the other hardware architectures, and it is surprising that the tensor processing units are still comparable to the other ones, but we are interrogating it further to understand how it scales with larger dimensional data sets. And finally, just to go over some of the key results from the study, the genes highlighted here are known to be regulated in diabetic kidney disease and implicate adverse metabolic effects, are known to be included in inflammatory pathways and also glucose metabolism. We observed the seven DKD markers, some of which we also saw in the previous slide, identified by the latent factors, in addition to four novel gene markers that are implicated in various injury based pathways from kidney literature. And some of those, such as metabolic pathways or biosynthesis of nucleotide sugars, are also seen in the study. The Hawk scores obtained from the latent factors and correlation analysis give or capture the higher order correlations among the gene, the cells, and the peaks. So, in summary, we developed this novel tensor based method, scHOCMO, to address challenges in single cell multi-omics data integration, creating a unified access of representation. The Hawk scores are capable of capturing the higher order correlations among genes, peaks, and cells in this case, and can be extended to include other modalities. The normalized append based stacking approach was found to be the most robust and efficient. GPU based execution was found to be robust compared to the other hardware architectures, and the latent factors were able to identify known as well as novel gene markers. For future directions, we are working with domain experts to enhance the biological interpretation of our results, as well as benchmarking on more than a trillion scale data set. And finally, I would like to thank Dr. Shamim Molla for giving me this opportunity, Molla Lab, Chow Lab, WashU RIS, and ISMB for having me here. Thank you. Happy to take questions.
Thank you. Thank you for the interesting talk. So I have maybe clarifications. So the reason why the scale is so big is because you're working at the level of single cells.
Right.
Have you looked into like pseudobulking, which is one way in which people try to reduce the number and also work with like, more so robust because you have a better signal. And my second question, if I may ask, is when you said annotate peaks to genes, what exactly do you mean by that? Was there a distance? Like what exactly are you doing there?
Oh, thank you. Those are great questions. Firstly, to answer that, how are we trying to, like, if we are trying to pseudo bulk, the rationale here was to look at each single cell readout and eventually extend the Hawk scores to, accommodate cell types, which would then kind of pseudo bulk or aggregate the readouts from different single cell resolution. But the idea is to keep the single cell resolution intact and then try to infer our cell identities or discover any novel gene markers. And by annotating genes to peaks, we mapped our peaks to like 200 base pair regions, and then we found the promoter regions mapping to those, and that's how we annotated the genes for that. No, not at this point, but definitely for future. Yeah. Thank you.